Hi everyone, this is Dr. LeBlanc with a video screen capture lecture of Eukaryotic transcription. Our learning goals are to understand how compartmentalization of transcription and translation affect the evolution of differences between eukaryotic and prokaryotic transcription, to understand the roles of the three different eukaryotic RNA polymerases, to understand the basic structure of RNA polymerase and its domain function, to understand the structure-function relationship of a eukaryotic promoter and its regulatory regions, to understand the difference between a cis-acting element and trans-acting factor, to understand the complexity and function of eukaryotic transcription factors involved in initiation and how they bind in a cooperative, cascading manner, to understand the role of the C-terminal domain of RNA polymerase II and the mediator complex, and to understand the two proposed models for messenger RNA transcription termination by polll 2. This picture shows a prokaryotic cell and a eukaryotic cell. And of course, the major difference between these two is the presence of a nucleus in a eukaryotic cell. Having a nucleus in the cell means that the process of transcription and translation occur in two different cellular compartments. In prokaryotes, transcription of the messenger RNA and translation can actually occur at the same time and in the same space. As soon as a messenger RNA is transcribed from its gene on the chromosome, immediately ribosomes bind and start to produce protein. As a consequence, messenger RNA in prokaryotes is very short-lived. It has a very short half-life on the order of minutes. As soon as the gene is finished being transcribed and the proteins have been translated from this messenger RNA, the RNA degrades. It degrades rapidly back into nucleotides, and in order to produce another messenger RNA, you have to start the whole process of transcription all over again. But RNA is usually very short-lived in prokaryotes. In eukaryotes, however, RNA has to last long enough to make it from the nucleus and out into the cytoplasm, a process which can take many more minutes. In addition, in some eukaryotic cells, messenger RNA is stored for many months before it is translated. Messenger RNA in eukaryotes has to be much more stable. One of the major areas of the messenger RNA that has to be protected is the ends, both the 5' and the 3' end, and there are specific modifications that occur on those ends, and we're going to talk about that a little bit more in the next video capture lecture. But the process of transcribing RNA in prokaryotes and eukaryotes is affected by the fact that the RNA has to last long enough to get out of the nucleus and into the cytoplasm to be translated. Here are two pictures of RNA polymerase from bacteria and from yeast. Yeast are eukaryotes. Notice that they're very similar. It's just that the eukaryotic RNA polymerases have a bit more substance to them. They're a little bit bigger, they have more amino acids, and they're a little bit more complex. But the overall structure is very, very similar, even to the point of having this alpha helix right here. In eukaryotes, there are three different RNA polymerases that transcribe three different sets of genes. There's RNA polymerase I, which transcribes all the ribosomal RNA genes except for the 5S ribosomal RNA. There's RNA polymerase II, which is the one we will spend most of our time talking about. It transcribes all the structural genes, in other words, all the messenger RNAs. And it transcribes some small nuclear RNA genes as well. And then there's RNA polymerase 3, which transcribes all the tRNA genes and the 5S ribosomal RNA. So 3 different RNA polymerases. And like I said before, POL2 is the one that we usually focus on the most. RNA polymerase in eukaryotes is a similar structure to prokaryotes. It has a cleft. The DNA enters here. A transcription bubble is formed where the DNA is unwound or separated into 2 strands. One of the strands acts as a template and the other is the coding strand. POL2 transcripts, or eukaryotic structural genes, have a similar structure to prokaryotic genes. The +1 nucleotide is also the first nucleotide of the messenger RNA. There is a core promoter with a Tata box at -25. And then there's the transcriptional start site. This is the sequence of nucleotides at the Tata box, the consensus sequence. And this is the sequence of nucleotides surrounding the transcriptional start site. Upstream from the Tata box is a common location for many different regulatory elements. When we talk about transcription and turning transcription on or regulating gene transcription, there are 2 types of parts to that process that we often refer to. One are cis-acting elements. These are actual DNA sequences. They are part of the genes. The other are transacting factors or regulatory proteins. The regulatory proteins bind to the cis-acting elements. So, for example, the Tata box is a DNA sequence. It is a cis-acting element. And we have Tata binding proteins, which are regulatory factors or transacting factors or transcription factors that bind to the Tata box. There are many proteins involved in the initiation of transcription of RNA polymerase II genes in eukaryotes. This is a partial list of some of the proteins involved. I don't want you to memorize all of these factors, but I do want you to understand that they bind to the DNA, that they bind in a specific order, and that they have different functions, and each of those functions is important to get RNA polymerase to initiate transcription, to start transcription. Okay, let's look at a couple of these transcription factors to understand what kinds of roles are necessary to get transcription to start. TF2D is the major first initiation transcription factor that binds to the promoter region. It is composed of a Tata binding protein, TBP, and other Tata binding protein associated factors. It recognizes that Tata box of eukaryotic protein-encoding gene promoters and helps to position RNA polymerase II at the right, correct start site so that it transcribes the right nucleotide as the first nucleotide. TF2B binds to TF2D, and then RNA polymerase binds to the core promoter. Okay? So TF2D with the Tata binding protein is necessary to bind first. TF2B binds to this and acts as an intermediary and gets RNA polymerase to bind to it. There's TF2F, TF2E, and TF2H, which is a very interesting protein. It's one of the final protein complexes that bind to the initiation complex. It has a multi-subunit protein and has multiple roles. It acts as a helicase to form that open complex. It also acts as a phosphorylase, and it adds phosphate groups to the carboxy terminal domain, or the CTD, of RNA polymerase II. And when that happens, it releases TF2B, allowing RNA polymerase II to proceed to the elongation phase. In other words, it promotes the promoter escape of RNA polymerase II. Let's see how these things bind to the DNA, to each other, to RNA polymerase II to get transcription to initiate. The TF2D binds to the Tata box. The Tata box is a cis element. It's part of the DNA. TF2D is a complex of proteins that includes the Tata binding protein and the Tata binding protein associated factors, or TAFs. A very interesting fact about Tata binding protein is that it is one of the very few DNA binding proteins that actually contacts the helix in the minor groove. Okay, after this happens, then TF2B comes along and binds to TF2D. TF2B is then going to recruit RNA polymerase II and another factor. Then TF2H binds to RNA polymerase II to form a preinitiation, or the closed complex. Here's TF2H right here. Okay, TF2H acts as a helicase to form the open complex. TF2H also phosphorylates the C-terminal domain, the very tip of RNA polymerase II. That's down here. When this is phosphorylated, when phosphate groups are added down here, some of these other factors are released and the promoter escapes, the RNA polymerase escapes the promoter, and elongation can commence. So we have a pre-initiation complex, which is a closed complex. TF2H, and it starts that helicase function. It starts to open up the DNA, break those hydrogen bonds to form that transcription bubble. And then all of these other factors are released after TF2H adds the phosphate groups to the C-terminal domain of RNA polymerase II. And now RNA pol II can proceed to the elongation stage. This C-terminal domain of RNA polymerase II is very, very important. These phosphate groups that are added and removed in different patterns during the process of RNA transcription are very important, and we will come back to this again. This image shows that RNA polymerase II itself, TF2D, TF2B, TF2F, TF2E, TF2H, and some of these other complexes, the mediator complex and the SWI/SNF complex, etc., are all made up of many different polypeptides that are all together in different complexes. So, for example, TF2D has a Tata binding protein and a whole bunch of other Tata binding protein associated factors to it. And altogether, this makes up TF2D. TF2H has many subunits as well. Okay, elongation occurs by simply adding one nucleotide after another, similar to what goes on in prokaryotes. Formation of the phosphodiester bonds as you add nucleotides onto the growing 3' end of the messenger RNA. There are two models of transcription termination in eukaryotes. When RNA polymerase II transcribes a gene, it continues until it reaches a polyadenylation signal. We're going to learn about what the polyadenylation signal actually means when we talk about RNA processing. But RNA polymerase goes past the polyadenylation signal, and when it does that, the messenger RNA is cleaved just beyond the polyA sequence. After that, many A's get added onto the 3' end. But how does the RNA polymerase actually come off the DNA template? Well, there are two models. The first is an allosteric model that states that after you cut this RNA and leave the 3' end near the polyadenylation signal here, this cis-acting element, that the proteins that are lost from the transcriptional complex cause it to dissociate. So they cause a conformational change, and that conformational change, that allosteric change from a distant site, causes RNA polymerase to fall off and the transcription bubble to be lost. And this short little transcript that's not a messenger RNA, it will be rapidly degraded, and RNA polymerase is free to start transcribing another gene. The torpedo model states that after the messenger RNA is cut off from this transcribing RNA polymerase, that there is an exonuclease that comes in and chews up this RNA that is still attached to the RNA polymerase and the transcription bubble, and that once this happens, because there's no more RNA, not enough RNA left here associated with the RNA polymerase and the transcription bubble, that everything falls apart as well. There's research ongoing right now to see which of these two models has more evidence to support it. So in summary, the compartmentalization of transcription and translation necessitates the modification of messenger RNA to increase its stability. Prokaryotic messenger RNA has a very short half-life. Eukaryotic RNA polymerase 1 transcribes the larger ribosomal RNAs, Pol II, the messenger RNAs, Pol III, the small ribosomal RNAs, tRNAs, and other small RNAs. RNA polymerase has separate domains for catalysis, DNA helicase, NTP binding and entry and clamping onto DNA. Eukaryotic core promoters have a TATA box, which is a cis element, to which the TATA binding protein binds. Transcription start sites have a loosely defined consensus sequence. The first nucleotide transcribed is designated as nucleotide +1. TF2D, transcription factor 2D, is the first transacting factor that binds during transcription initiation. TF2D is a complex of proteins that includes the TATA binding protein and several TATA binding protein associated factors, or TAFs. When the C-terminal domain of RNA polymerase 2 is phosphorylated, its function changes. The mediator is a large complex of proteins that regulates the expression of genes by interacting with both the TATA box binding transcription factors and others that bind at a distance. This was introduced in this lecture, but we will come back to this in subsequent lectures. There are two different models to describe messenger RNA transcription termination by Pol II: allosteric model, loss of transcription complex subunits, and the torpedo model, degradation of mRNA and dissociation of the transcription complex from the template.